Hello lovelies, in this video the brilliant Dr. Webblitz is going to be taking you through your WJEC Separate Science Biology Paper 2 for the foundation tier. Now if you haven't had a go at this paper already, what I want you to do is to pause after the question comes up, have a go at it yourself on a random bit of paper, play and then go through the answers with Dr. Webblitz together.
Hi everyone! In this video we're going to go through the WJEC Biology Separate Foundation Paper 2. You can see it here, hopefully you've downloaded it and attempted the paper, and we're going to go through each question step by step. I'm going to analyse the questions, highlight, annotate, help you to figure out what the question is asking, give you some general tips for doing that in the exam, give model answers, and then explain the mark schemes as well, so you can use this to help you mark. So if you haven't already marked it, then you can mark along with me. If you have, then I'll help you by explaining the mark scheme so you can see what you would and wouldn't have got marks for and why. Okay, off we go. So question 1. Stem cell cloning can be used to quickly produce clones of plants. So the first question says, describe the key features of the following. So key features here, what it means is if it's asking for the key features of something, it's asking for things that make them unique, identifiable features, things that you know that you can describe about that means that you can recognise it and it's only relevant to that, whatever it is you're describing. So our first one is embryonic stem cells. So embryonic stem cells are stem cells that come from embryos, and they can differentiate into any type of specialised animal cell. So they can become any type of animal cell in the body of that organism. Then we have adult animal stem cells. So these are the ones that you find in bone marrow. So they're the ones that you have after a certain point. Your embryonic stem cells stop being able to differentiate because you're not an embryo anymore. You start growing into a fetus and a baby, and so then from that point on, all the stem cells that are left in your body are adult stem cells, and they can only differentiate into some or a few types of cells. And then lastly we have plant stem cells, and you'll notice here that this one is worth 2 marks as opposed to 1 mark. So that means we have to say 2 things to get the 2 marks. So plant stem cells are like embryonic stem cells in that they are able to differentiate into any type of plant cell. But the unique thing about plant stem cells is, unlike embryonic stem cells, they retain that ability throughout the whole life of the plant. So it's not just when the plant is young that they have those stem cells, they have them throughout their whole life, which is how we can do plant cloning and things like that. Okay, so name the part of the plant where the stem cells are found. That's obviously just a fact that we have to know. So hopefully we know we find them in the tips of roots and shoots in what we call the meristem tissue. Part C. Give an example of tissue damage that stem cells could be used to treat in humans. Okay, so this is asking us, what could we inject stem cells? What tissue could be damaged that we could inject stem cells and it would help cure the disease? So hopefully you've looked at some examples of things that stem cells can be used to treat. One of the main ones that you'll probably have looked at is paralysis. So what tissue is damaged that causes paralysis is really what this question is asking. So it is nerve tissue that gets damaged which causes paralysis, because no nervous signals can then pass along. And so that's why you can't move certain parts of your body, which is what paralysis is. So the tissue that's been damaged in this case is nervous tissue. So we're going to put that, and obviously you can potentially inject stem cells which could become new nerve cells and become new nervous tissue, and that could fix the problem. Okay, so we have our mark scheme for question 1. As we've said, the main things really for those first 3 points is the differences between them. So embryonic cells becoming any type of cell, adult stem cells only some types or a few types, and then plant stem cells becoming any type of plant cell, but also being able to differentiate through the whole of the plant's life, or basically having that ability through the whole of the plant's life. That gets you the 2nd mark for the plant stem cells. We talked about nervous tissue and paralysis. You could have talked about spinal cord tissue or brain tissue as well, which can be linked to paralysis, or you can talk about the pancreas. So in type 1 diabetes, the pancreas tissue is damaged, it's not working, it's not producing insulin. So it might be that you would talk about that for type 1 diabetes as the tissue that's been damaged there. It's about making sure you're talking about the tissue that's damaged, because that's what the question's asking.
Question 2. Chlamydia is a disease that is transmitted during sex.
It can cause yellow or green discharge and pain. Select the type of pathogen that leads to chlamydia. So we've got options here. Think about when you've looked through this list, what examples have you learned for each of these? So protist will be malaria, virus will be probably HIV, and fungi is normally athlete's foot for humans. So bacteria is our only other option if you didn't remember that chlamydia was caused by bacteria. On to part B. So give one method that can prevent a person from being infected with chlamydia. Well, there's a clue at the start of the question where it says that it's transmitted during sex. So if you didn't remember that that's how chlamydia was spread, then this is going to help you. So one way to prevent the spread, if it's a sexually transmitted infection, there's only one method that you will have learned, which will be condoms, or a barrier method of contraception, such as condoms. Part C. When a pathogen enters the body, the immune system tries to destroy it. Match up the correct ways the white blood cells defend against pathogens. Draw one line from each white blood cell. Okay, so when we say defend against pathogens, it's how they destroy the pathogens. How do they find them and then break them down and remove them from the body? Pathogens, remember, are microorganisms like bacteria and viruses. So we've got two types of white blood cells. We've got phagocytes and lymphocytes, and we need to know which one is which and which way they end up removing sort of bacteria or microorganisms from the body. So phagocytes, there's a clue in the name. They go through phagocytosis, which is engulfing or surrounding and digesting pathogens. So I would do this one first because it's easier, and go down the multiple choice every single time and make sure you check which one is which. So it's not produce antibiotics, engulfing pathogens, yes, so it is that one. So we can stop there and join our one line up to there. We're really clear, we're drawing one constant line, it's touching both boxes, and we're not gonna make it ambiguous. So we're gonna make it very clear which line is which. Lymphocytes, we can go through what's left. So antibiotics, no, they kill bacteria, but your body doesn't produce them, they're drugs. Vaccines, again, you get injected with that, it's not something your white blood cells do. Produce antibodies, yes. That is something our white blood cells do, and it must be lymphocytes because they're not phagocytes. That's the other type of white blood cell, so we can draw our other line. Doesn't have to be straight with a ruler. If you had a straight ruler in the exam and you could draw a straight line, that would be great, but it doesn't have to be. It just has to be clear, and make sure you've only got one line coming from each box, and make sure if you do make any mistakes, you really make it clear what your actual answer should be. Part D. Chlamydia can be treated with prescription drugs. What drug can be used to treat chlamydia? Okay, well, way back at the start of the question in part A, which seems like a while ago, we had to choose what pathogen causes chlamydia, and it is bacteria. So it's not antivirals. Antibodies are something that white blood cells make, and painkillers treat pain. They don't actually kill pathogens. So it has to be antibiotics because they're the only ones that kill bacteria. Now, obviously, that relied on us knowing that chlamydia was caused by a bacteria, but hopefully we knew that. Explain why the same drug cannot be used to cure an infection caused by a virus. So this is basically asking why won't antibiotics kill or treat a viral infection or a virus. So antibiotics work to kill bacteria, but the bacteria that they're killing are outside of cells. So the difference between that and a virus then is that viruses replicate and live inside your body cells. So they're two different things. The antibiotics kill bacteria outside of cells, which means they cannot get inside your cells. They don't work inside cells, and that's where the virus particles are. So that means they can't kill them because the virus particles are inside a cell, and the bacteria, the antibiotics can't get there because they can't actually get inside cells to kill the virus particles. So the mark scheme for question 2, so we've got the bacteria, which is the right pathogen. We've got condoms. It will accept if you put a barrier form of contraception, that's fine. You've got obviously the 2 right answers, 1 mark for each for the white blood cells. Then you've got antibiotics, and then we've got that 2 marker, so we've got to say that viruses replicate inside cells, and then antibiotics can't enter cells, or they don't work inside cells, or the antibiotics don't kill human cells. Because if you killed a cell that contained a virus, then the virus would be killed as well. So any of those kind of options for that second mark.
Okay, on to question 3.
The image shows 3 dogs. They are all members of the same species. What is the definition of a species? So this is a 2-mark question. It's just a definition question. Hopefully you know the definition of a species, but just to be clear that we've got to have enough detail in there to get the 2 marks. So we need to say that they're similar organisms and that they can breed, but we need to make sure we also say they can breed to produce fertile offspring. Because remember, things like lions and tigers are similar. They can breed, but their offspring are not fertile, which is why lions and tigers are therefore different species. So a group of similar organisms that can breed to produce fertile offspring. The table below shows the classification for dogs. Complete the table. Okay, so we've got a classification table or a taxonomy table, and we need to fill in those gaps for the names of the groupings. So hopefully you can try and remember the sequence. I like to use king, prawn, curry, or fat, greasy sausages as a way of remembering the letters in order. And then we just need to remember what those letters stand for. So the K is for kingdom, the O is for order, and the S is for species. And then obviously we have phylum, class, family, and genus already done for us. So we just need to make sure we've put the right words in the right order. So yeah, use that and like an analogy to kind of remember the K, P, C, O, F, G, S. Just remember the right order, and then it's just about remembering what those letters stand for. Give the binomial name for dogs. Use the table above. So hopefully we remember the binomial name means two name or two-part name, and it comes from using the genus and the species names from the table or from their classification. And so the binomial name for dogs, its Latin name, is going to be Canis familiaris, and we need to copy it exactly as it is in the table. So capital C for Canis and a lowercase f for familiaris, because you always leave the species name as a lowercase.
Give one reason why binomial names instead of conventional names are used by scientists. Okay, so this is asking why we use that like double Latin name instead of using a name like dog. So why do we have a Latin name for dogs instead of just calling them dogs? I think the easiest way that I like to think about explaining this is if you look at dog in different languages all over the world, all of the words for dog are different. But if you use the Latin name, doesn't matter where you are in the world, the Latin name is going to be recognised by everyone as meaning dog. So that's one of the main reasons we have this Latin name classification system, because it means that everyone all over the world, regardless of language, will know what you're referring to, and we're all talking about the same thing. The traditional classification system only contained two kingdoms. Who developed this system of classification? So we've got Charles Darwin, Carl Linnaeus, Carl Woese, or John Baptiste Lamarck. So it's not Charles Darwin because he's to do with natural selection and evolution, same with Lamarck. You've got the two Carls are the classification Carls, as I like to call them. So you've got old classification system, which is Carl Linnaeus, and the new updated version, which is Carl Woese. So the old original system is Carl Linnaeus. Part F. So the new three-domain system with 5 kingdoms was introduced in the 1990s. Explain why classification changed to the three-domain system. So why did we need to change it, but also sort of what was the reason for the updating? So there must have been something that caused the change or why we needed to update the system. And it was mainly to split the prokaryotes into the archaea and the bacteria into 2 groups, which made these new 3 domains and ended up adding a new kingdom because we split prokaryotes, which was 1 kingdom, into 2. We need a reason why that was done though. So why did we split them? Well, we had DNA analysis and electron microscopes and new technologies and techniques which basically gave us evidence that showed that they weren't closely related enough to be in the same group, and so they needed to be split. So I would go with DNA analysis because it's obviously the most modern technique that we have, that we use to do this. So the DNA analysis showed that they were too different, or they were different enough that they needed to be separated. So that's why we had to change the system. So the mark scheme for question 3, we've got our kind of 2-parter definition for a species, which we just have to know. So similar organisms that can breed together. Instead of similar, if you put same number of chromosomes, or yeah, that's basically it. You can only really say that. Can't say have the same number of genes because that doesn't mean that they are similar. And then you can't really say mate. You need to try and say breed because that suggests obviously they're sexually reproducing and actually producing offspring, and then the offspring have to be fertile. You need that word in there to get the second mark. For the words missing in the table, you get 1 mark for one of them correct, and then 2 marks for all 3 correct, and obviously they have to be in this exact order. You can't just have those words but in the wrong places. Careful when copying over the binomial name. You need a capital letter for Canis, and you need a no capital or a lowercase f for familiaris. You are just copying, so it should be exactly spelt correctly and everything exactly the same as where you're copying it from, so just be careful. for the kind of name that we said. So you can either have the same name used by everyone all over the world, which is what I was saying regardless of language, or you can talk about the fact that it's useful to have that binomial name because the first part or the genus part gives an indication about evolutionary relationships. So you can kind of use it to think about other animals that also share that genus, so what it might be related to. It helps to know which species are closely related to each other. Linnaeus, as I said, was the developer of the original classification model. Why did we need to update it to the new one? Well, we had electron microscopes, which allowed us to see more detail, or DNA analysis techniques that developed over time. And then that allowed us to separate, or gave us evidence that said that we needed to separate the prokaryotes into two groups. If you didn't get any of those marks and you were just describing the difference between the two sort of systems, and you talked about the fact that there were now 3 domains that have archaea, prokaryota and bacteria in, then you can have 1 mark if you didn't mention anything else that was relevant in terms of getting the other marks. Question 4. The diagram below shows a cross-section of the eye. It is a sense organ and is part of the nervous system. It contains receptors connected to sensory neurons. Okay, so we've got a diagram now and it's been labelled for us, which is quite nice. The first question says, in which part of the eye are the receptors located? So we've got a diagram and it's been labelled, so we're clearly going to need to talk about one of these labelled parts. Just remember, obviously, this is how light comes in through the eye, through the pupil, and then it hits the back of the eye. And so that's where the receptors are located, in the layer called the retina. Again, this is something we should know. Don't have to work it out from the diagram. It should be something that we've just been taught. Then what stimulus do those receptors detect? Well, obviously they detect light, we've done that, but remember that a stimulus is a change in the environment. So I would say, although they do detect light, what they're actually detecting is, so wavelength of light or light intensity, changes in the light intensity. That's the stimulus, isn't it? Light is there all the time. It's about the changing of the light or the differences in the light that it's detecting. But I think if you put light, you would still get the marks. We'll check in the mark scheme in a second. Then it asks us for 2 examples of automatic reflex actions. One must involve the eye. So this is something that students tend to struggle with, is thinking about what actually are examples of true reflex actions, like throwing a ball and catching it or jumping out the way of a moving car or something. None of those are reflex actions because they require you to use your brain to think about them. True reflex actions are not under conscious control. They happen without you thinking about them, and they are kind of involuntary. So one of the couple involving the eye that we should know is things like blinking. When something comes close to the eye, like flinching, you can't help that. If something comes close to your eye will just shut on a reflex action, or it will blink at least. And then the other one is your pupils dilating, so the changing in the contraction or the relaxation of the pupils depending on how much light there is, to kind of stop too much light entering your eye or allowing just enough light in to be able to see. That is also something that you don't think about. And then some other non-eye related ones are things like the knee-jerk response. When you're just below your knee on your shin gets hit, you can trigger a nervous response that is like your knee to like kick out. And something else is, so thinking about sneezing or coughing when something goes into your nose or you breathe something that aggravates and irritates your throat or your nasal passage. If the hairs and things inside are triggered, you will sneeze or cough, again, just as a reflex action to try and get rid of whatever's in your airway. So we need to pick 2 of these, and one must involve the eye at least. So you probably could do 2 that involve the eye, that's absolutely fine. I'm going to choose the knee-jerk reaction and then the pupil reaction to light intensity. Okay, so part B. One of the functions of the autonomic nervous system is to control reflex responses to danger. Suggest why reflex responses offer an evolutionary advantage to humans. Okay, so reflex responses, how do they allow us to survive and reproduce? That's what an evolutionary advantage is. It's an advantage over other organisms that's going to help you to survive and reproduce, and therefore it will help you evolve, or it'll help you survive and therefore adapt because you'll be able to pass on your genes, whereas other people who don't have this advantage might die out. How do reflex responses then help you to survive? Well, they're really fast, and like we said, they bypass the brain, they are not under conscious control, and so that helps us to that the response is so rapid, it protects us from harm, protects us from danger. So thinking about things like touching a hot pan and you jerking your hand away, touching something sharp, jerking your hand away, protects you from being injured or burned. That can obviously help us survive so that we're not going to get injured or burned in a way that then's going to stop us from being able to look after ourselves or hunt or whatever it is. So they're fast, and that helps keep us safe from harm or danger. So that's the evolutionary advantage. This is a suggest question. They are designed to be hard. They are designed to make you really, really think and kind of go beyond the specification a little bit, or link things together. So don't worry if you found this question hard. It's designed to be. So just talk about how fast they are, and then talk about the fact that that's going to keep us safe from harm.
Two students wanted to compare the function of their nervous system by testing their reaction time using a ruler drop test. Hopefully we've recognised this already as the required practical for paper 2. There aren't many required practicals for paper 2, so it's definitely worth learning the few that there are. So this is the one where we're like using ruler drops to test our reaction time. So they used the following method. Student A held the ruler just above student B's hand. It doesn't say which hand, so that's kind of important, and we'll talk about that as one of the variables in a minute. Student A let go of the ruler without warning, and student B had to catch it with their thumb as soon as possible. The reading on the ruler where the student's thumb was in centimetres was recorded to show how far the ruler fell before it was caught. They repeated this 5 more times, then swapped over to student A was catching. So they are measuring the number of centimetres that the ruler fell through their hand before they caught it, and they did repeat the experiment, which is good. It's important to repeat experiments, we should know, and it means that we've got some sort of reliable results. Okay, so the actual question says, state one factor the students could have controlled to make this investigation fair. Well, it's basically just asking what should be kept the same for both students. And so anything that should be controlled is something that can affect what you're measuring. So in this case, we need to think about all the things that can affect reaction time, and they're all the things that should be kept the same unless you are changing them. So they're not testing to see if there's any specific effect of tiredness or caffeine or anything in this practical. They're just looking to see what the difference is between the two students. So we need to think of things that can affect reaction time. So how tired you are, your age, your sex, how much practice you've had, how much caffeine you've had, whether you've used the same hand. So in this case, that's kind of linked to practice, but if you use a dominant hand, that's going to be stronger than a non-dominant hand, and you're used to catching things, so you'd expect your reaction time to maybe be faster with your dominant hand. So all of those can affect reaction time. I'm going to say the students should both use their non-dominant hand, but you could say they both need to be the same age, they both need to be the same sex, they both have to have not practiced before, so they're using their non-dominant hand, or they both had to make sure they hadn't had the same different amounts of caffeine that day. One had a coffee and one didn't, and that wouldn't be fair. So any of those that can affect reaction time we can use as a control factor.
After converting their ruler measurements to reaction times using a table, they obtain the following set of results. So hopefully if you've done this practical, you remember once you've got your result, you need to convert that to the reaction time in seconds. So they've got the following results. They want us to calculate the mean reaction time for student A. Now this is a 2-mark answer. So first of all, how do we calculate the mean? Well, we add up all the numbers and divide by the number of values. However, if you're calculating mean, you should know that you should do that, but only do that once you've removed any anomalies. So we need to take out any anomalous results and then add up all the numbers and divide them by the number of values. So I'm looking at the list for student A. So we've got 0.27, 0.4, 0.23, 0.24, and 0.22. Now the 0.4 is jumping out at me immediately as a potential anomaly because it's nearly double all of the other values. And if you look over to student B as well, their highest was 0.31. So it's way higher than any number on the table, whether it's for student A or student B. So I would say that this is most likely an anomaly, given how close in range all of the other numbers are. They're all in the 20s. I would say we remove that, and so then we calculate 0.27 plus 0.23 plus 0.24 plus 0.22. That gives us a total of 0.96. And then we're dividing it by 4 because we only had 4 values, so that gives us 0.24. Give one conclusion you can make from these results. If you're looking for conclusions, especially in a table, then you need to look for patterns or differences that you can talk about. So there's a pattern that I can see, which is that for both students, the reaction time decreases as they have more goes. So it decreases with practice, which might be something that you looked at when you did your reaction time. And also we have a difference. So student A's mean reaction time is lower than student B, so they have a faster reaction time than student B. So there's a difference and there's a pattern. I'm going to go for the difference because that's kind of what they were investigating, was they were comparing their reaction time.
Okay, so the marks given for question 4. So as I said, it's not sort of good enough to say just light. The sort of stimulus they respond to light intensity or changes in light intensity. Because that's what if you think about your pupils dilating and reacting, they're responding to the intensity of the light or the amount of light. So they're not just responding to light on its own. So that is important for you to kind of make sure that you've put that in there for that answer. We went through those options for the reflex responses. You can have any of those, and also the one I was talking about with the sneezing, that should be fine as well. The fact that they are fast or rapid, you needed for the first mark to say why that they're helpful to stop you from being injured or harmed, or to prevent you from being damaged, and then ultimately increases the chance of your survival, which is what we said was linked to this idea of why it's an evolutionary adaptation. Any one of these control points can get you a mark. You need to think about all of the things that can affect reflex reactions. So practice, the sex of the person, the age of the person, the same time of day, which is going to figure out or be linked to how tired they are. So the same amount of sleep is also linked to how tired they are. The same amount of practice, which is kind of linked to the dominant hand, because obviously any hand you use more is going to have sort of be better skilled at catching things. So that's kind of linked in, but you can have either of those. And then weight of the ruler. So weight or mass of the ruler is fine, because that will affect kind of the drop and the kind of how hard or easy it is to catch. But you can not just say the same ruler. You need to say what about the ruler needs to be the same, because what about the ruler could affect the experiment. I would avoid, for most practicals, I would avoid talking about equipment when talking about control variables. Very, 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 very rarely is equipment ever going to be a control variable, or is ever going to affect the experiment to the point that the equipment needs to be controlled. It's most likely going to be something else that you can easily figure out that is actually more likely to affect the experiment than the equipment. So think of other things, scientific answers first, before you start thinking about, oh, well, the equipment should be kept the same. There are reasons why it should be, but most of the time, there's normally other reasons you can think of. If you try to work out the number for 4D and you didn't get the right answer, but you did do the working in the same way that I did, then you can get the mark, or you can have 1 out of 2. And then for E, remember we're saying about what conclusion that we made. So ideally, we need to be talking about faster reactions, because that's what we're looking at here. So student A has faster reactions than student B, or student A caught the ruler faster than student B. You need that kind of comparative statement here. So student A versus student B, what does the data show is the difference between them? Or, because we recognised it as a pattern, that reaction time decreased with practice. So the more tries they did, as they were doing more and more tries, they were getting better. So that's another pattern that you identified. So that can also be a conclusion. Question 5. Select the process by which bacteria divide. Tick one box only. Okay, so bacteria divide by binary fission. They don't divide by mitosis. Meiosis is our first option here. That's to do with making gametes, so sperm and eggs, so it's not that. Phagocytosis we've already talked about, is that kind of engulfing of bacteria. And then plasmids are just the loops of DNA that we find in bacteria. So has to be binary fission. Again, this is just one of those pieces of content that we have to know. students set up a practical to investigate the effects of antiseptics on bacterial growth, as shown in figure 2 below. They placed discs that had been soaked in antiseptic onto an agar plate that was covered in bacteria. So hopefully we recognise this as a required practical about growing bacteria, and we're doing it with antiseptics. So you might have heard about doing it with antibiotics, but antiseptics they're just chemicals that kill bacteria on the surface. So they're not something you put inside you, but things like bleach and other cleaners that you would use to kill bacteria. So hopefully you recognise the practical, and then what it says, the next part says, select the reason for each of the following stages in the method. So this is a common thing they do with required practical questions, is where they're asking you why it's important each of the steps have been done that way, or why is it important that that method step gets done in that way. It's normally because it's going to affect either the results or the experiment or something. The first one is labelling the plate on the bottom of the agar dish. Again, let's go through the multiple choice. So you can't write on the lid. That's not true. You can absolutely write on the lid, so it's not that. To avoid contamination, writing on the lid or underneath the lid is not going to contaminate it, so that's not true. It would look messy. It could look messy if your handwriting was messy, but realistically, that's not going to affect the results of the experiment. Whereas to ensure labels stay in the correct place could affect the results, because if your labels move around and you don't know which disc is which, that's a problem. Next one, opening the lid away from you. Again, to let other people see what you're doing, no, that's not true. That would be silly. No one else needs to see what you're doing. To ensure the lid stays in the correct place, again, we've established that we shouldn't be writing on the lid anyway. To allow oxygen to get in, well, when you shut the lid of a Petri dish, it shouldn't be airtight. You've not sealed it in, so air can get in anyway. So to avoid contamination, is that going to could potentially affect our results? Yes, because if you contaminate it with bacteria from breathing on it, then that could cause a problem with the results, because we'd be growing bacteria other than the bacteria we'd be planning on growing. So you can see how when you work through the multiple choice, you can narrow it down using kind of common sense, or figure out things that definitely aren't right, and then think about, when you have decided on your answer, think about, is that going to affect the experiment? Is it going to cause an issue? So part II, so using forceps to add the discs. So again, we need to think about, why would you use forceps, so the metal tweezers, why would you use them instead of using your fingers? What can you do to the forceps? Well, it would prevent you from dropping them. It could, but it's unlikely. Is it to prevent it looking messy? Again, that doesn't matter. Is it to work more quickly? Again, doesn't matter. To allow for more precise placement, so making sure they stay far apart from each other, yes, that makes sense. That would help you to do that. And then lastly, no multiple choice this time, explain the reason for sealing the lid with small bits of tape. So why would you not seal it all the way around with one long piece of tape and stick it down? Well, if we did that, then we would have sealed it so that no oxygen could get in. And obviously that's a problem, because the bacteria need oxygen to grow, because otherwise they won't be able to respire, they won't be able to replicate, reproduce, and we're looking for that because we need them to grow to know whether the antiseptic has killed them or stopped them growing or not. So the plate needs to be partially sealed to let oxygen in for the bacteria to grow. So, or you could say the plate shouldn't be fully sealed, or only needs to be sealed partially, however you want to say that, that's fine. So the bacterial plate was incubated at 25 degrees C for 36 hours. Assume there were 1,000 bacterial cells when the plate started, and then the time it takes for bacterium to divide is 45 minutes. So that's the time it takes to double. So one bacterium will turn to 2 after 45 minutes, and that's all of them. And we're starting with 1,000. So it wants us to work out how many bacterial cells there would be after 4 hours and 30 minutes. We just need to work it out in the steps that they've given us. So the first thing it wants us to do is to determine how many times the bacteria will divide in 4 hours and 30 minutes by calculating how many lots of 45 minutes there are in 4 hours and 30 minutes, which is nice of them to help us out. So the first thing we need to do is convert the 4 hours and 30 minutes into minutes. So there's 60 minutes in an hour, so 4 times 60 gets us 240, and then we've got the 30 minutes to add on. So that gives us 270, and then we do 270 divided by 45 to see how many times it goes into it, and we get 6. So there are 6 divisions happening. So all the bacteria in the 1,000 bacteria there are will divide 6 times in that 4 hours and 30 minutes. So it then says, state what happens to the number of bacteria every time they divide. Well, the number of bacteria is going to double, because we've said they're all going to go from being 1 split into 2.
And then lastly, calculate the number of bacteria at the end of 4 hours and 30 minutes. Well, we just know we need to take 1,000 and double it 6 times. So we can just write this out. If you've got space for working, hopefully you can. If not, ask for a spare piece of paper if you think you're not going to take up much space. Or if you want to just do it on the calculator, that's fine. Just be careful and make sure that you write down some working if you can. So start with 1,000 and double. So we get to 2,000, double that, gets 4,000, double that, gets us 8,000, double that, gets us 16,000, double that, gets us 32,000, and one more time, double that, gets us 64,000. Now, we have to make sure that we've done the right number, because a lot of people sometimes just count the numbers and think they've done 6 doublings, but actually they've only done 5. You need to count the arrows. So if you write it out like this, you should do the arrows. So 1, 2, 3, 4, 5, 6 arrows means 6 doubles. We've got it right. Okay? To determine the most effective antiseptic, a student wanted to measure the clear zone around the top 2 discs on the plate. To describe how to determine the area of the clear zone. So we're just looking at those top 2 that've got that clear zone of bacteria around them, and it wants us to describe how to determine the area. So it wants like method steps, so literally methods, kind of what you would do step by step in order, and how you would measure and then calculate the area of those kind of circles that we've got. So we can't just measure it once because that's not very reliable, so we need to try and make sure we measure it twice. But you measure it once, the kind of length, the diameter of that circle, and then you're going to go 90 degrees or right angles to measure it again, and then we can take a mean of those 2 numbers. So we've taken like the 2 widest parts, we've done it on 2 different angles, and then we can calculate a mean from that. So once you've got the mean diameter, we can calculate the area of a circle. Hopefully we should know that the area of a circle is pi, worked out by pi r squared from maths. If you can't remember that, I like to remember the rhyme, when you need a hole repaired, what you need is pi r squared. So a hole repaired means you need a hole, you need to find the area of the hole you need to fill in, and so you use pi r squared. And so with the diameter we can do that because you can half the diameter to get the radius. So this is a bit of a maths question as well. But realistically, as long as you've described how you would measure it, how you would calculate the mean, and then say that we'd use that mean diameter to calculate the area, that's really all we need here. So measure the diameter of the clear zone around each disc twice at right angles to each other, calculate the mean diameter for each disc, or the diameter of the clear zone for each disc, and then using the mean diameter, calculate the area using pi r squared.
Part E. The students wanted to stop the bacteria on their plates from growing. The students disagreed on how they should store them. Student A said, "I will put my plate in the fridge because I don't want my bacteria to die." Explain if you think student A is correct and what the students should do to stop the growth on their plates. So for 3 marks, there's quite a lot we can say here. So we're going to have to say whether we think A is correct, and we need to explain why or why not. And then it says what should the students do. So then we have to suggest what they should do in order to stop the growth on their plates. Student A is saying that they want to put their plate in the fridge, but that won't stop the bacteria growth. It will slow it down, but it won't stop it. And then she said, "I don't want my bacteria to die." So that's suggesting that they need a coldish temperature, but not too cold, so that they don't die. That's not true. So the bacteria, if you put them in the freezer, the cold temperatures will be enough to stop the growth, but they won't die. They just go into like a dormant phase. So we need to say that A is wrong, because being in the fridge won't stop the growth, it will just slow it down, and that they're wrong because the bacteria won't die if you put them in the freezer, but they should use the freezer because it will stop the growth. So that covers everything, explaining why we think student A is wrong and then suggesting what they should do instead. So student A is wrong because the fridge will not be cold enough to stop the growth of bacteria. They should put the plates in the freezer to stop growth. It will not kill the bacteria. Okay, so for the mark scheme for 5A, obviously we had binary fission, which was our choice point, and then we've got our multiple choice options for the methods and why we do those methods for the practical. Then we've got kind of our reasoning why we don't seal up and tape up our plates, and you've got options here that you can have any 2 from. So because it will stop the lid from falling off, which is obviously helpful. So it's not fully sealed to prevent anaerobic conditions, and then you can follow that up by saying if we grow things in anaerobic conditions, they're more likely to be harmful or pathogenic. If we sealed it up entirely, the bacteria wouldn't have oxygen and so they wouldn't be able to grow. Are perfectly valid, you just need to pick 2. So we had to work out that there were 6 divisions. Binary fission, we should know that that means that the number doubles every time. And then we've got a couple of ways of working out that maths. You can either use the start number times 2 to the power of the number of divisions. So 1 is our start number times 2 to the power of 6. Or you can literally write it out properly. So you can literally say the minutes, double the minutes, and then double the number of bacteria. Then we've got our method steps. There are only 4 points and there are 4 marks, so you have to say it exactly right. You've got to measure the diameter. You've got to measure it in 2 different places at 90 degrees or perpendicular to each other. Either of those is fine. Calculate the mean and then calculate the area. Ideally using pi r squared would be for that last mark because it didn't tell you how to calculate the area. So yeah, using that formula. And then as we said, student A is wrong because we said that freezing won't kill the bacteria, or that you could say the way around, the fridge wouldn't stop the growth of their bacteria, whereas they should store their plates in the freezer. So there's kind of those 3 points there. They're wrong, why they're wrong, and then what they should be doing instead.
Okay, on to question 6.
The control of blood glucose in the human body is important for the maintenance of proper blood composition and general health and well-being. The concentration of glucose in the blood is controlled by a specific organ. Identify and label this organ on the diagram below. So this is a long question, but basically it's asking what organ controls or coordinates blood glucose levels in the blood. The diagram that you've got in order to choose from is the endocrine system. So it must be a gland that we are looking for here. So although we know or should know that the pancreas and the liver are the 2 main organs involved in blood glucose control, only the pancreas is a gland, so therefore, and the liver is definitely not on this diagram. Even by process of elimination, if you weren't sure, then you should be able to tell that it should be the pancreas. So then it's about identifying the pancreas on the diagram and labelling it. Now, I'm circling it here so that you can see what I'm pointing at, but I would potentially just try and draw a nice straight line, touching the pancreas, make sure the line is touching it, and then label it and make sure obviously you spelled pancreas correctly and written it clearly. Okay, part B. If the level of glucose in the blood becomes too high, this organ releases a specific hormone. Name this hormone and outline how it lowers blood glucose concentration. So again, this question is quite long, but basically what hormone lowers blood glucose levels and how does it do that? So hopefully you may have heard this before. If you haven't heard this before, this is my way of remembering which hormone in blood glucose control does what. How to remember which hormone you need to choose for which scenario. So my way of doing it is to remember the phrase, when glucose is gone, you need glucagon. So when gluc is gone, you need glucagon. So that means when the blood sugar level is really low, you need glucagon to bring it back up. And if you remember that phrase, then you know that insulin is the opposite. So therefore, insulin is the hormone that gets released when there's too much glucose. And that's how I remember the way around to do it. So in this case, it's asking us what happens if blood glucose levels become too high. So which one is going to lower it? That's going to be insulin. So we need to say it's insulin, and then we need to be able to say how does it lower the blood glucose levels. So it has to remove glucose from the blood into cells, and it does that because when it goes into the cells, it converts it to glycogen. So this is just an outline question, so it's not really complicated to explain exactly the steps and what happens, and we don't have to mention the liver necessarily. It's 3 marks, so we're going to get 1 mark for saying insulin, and then we're going to get 2 marks for our outline. So it causes blood glucose levels to decrease, or it causes glucose to move out of the blood and into cells, and that's because in the cells it's then converted to glycogen. So insulin causes glucose to move out of the blood and into cells to be converted to glycogen.
Part C. Complete the table to show the differences between the causes and treatment of the 2 types of diabetes. Okay, so we need to say what causes type 1 diabetes, what causes type 2 diabetes, and then the treatment for type 1 diabetes and the treatment for type 2 diabetes. And obviously, given that it's asking us to talk about the differences, we need to make sure we can show what is different between them. So the cause, remember, is like, what is it? What's making it happen? And then the treatment is how we cure it or how we treat it with medicine or drugs of some kind. Again, focus on the differences, make sure we know and are clear about what the differences are between these two types of diabetes. So type 1 diabetes is where the pancreas is not producing insulin. So you have no insulin and you're not producing it. Whereas type 2 is that you're producing insulin, but it's not being recognised, or the body's not responding to it. It's not being detected by receptors in the body. So it's almost like it's not there, but it's being produced. It's just that it's not being detected by cells. Then having to think about what you need to do to treat that. So if you don't have any insulin, if you have type 1, you will need to inject insulin in order to have it in the body to do its job. Whereas for type 2, it's often due to things like obesity and fat buildup up around the organs, including the pancreas. So diet and exercise is what is used to start with as a treatment for type 2 diabetes. Okay, so you just need to make sure we put those right things in those right boxes in order for us to get the marks. Mark scheme for question 6. There's 2 marks, one for being able to label it as the pancreas and one for being able to kind of locate that correct organ which you are then going to label. So it is this organ here, the kind of leaf-shaped organ. So if you pointed it with a line or you circled it, as long as you're pointing and circling that organ, that's fine and you can get the marks. We need to talk about how insulin is removed from the blood. How insulin removes glucose from the blood. So the glucose moves from the blood into cells and the excess glucose gets converted into glycogen. So it gets turned into a different molecule and stored, and that's where the glucose goes. Again, that is a process. It's something that we need to know. Then we have our differences. So these have been written out rather than in the box, but they must be in the correct boxes for each type. So how we had them. So making sure that you've got no insulin one side, insulin not being responded to one side. Then you've got the insulin injections one side and the diet and exercise the other side. Question 7. Match up the following terms to their correct definitions. Okay, so we have some words which we need to match up to their definitions. I would say spend a bit of time for each of the words thinking about what you think they mean and getting your kind of notes down about your definition, and then use that to help you determine which statement is the closest to that from the definition they give, because it's not always going to be identical to the definition wordings that you've learned. So dominant is where we only need one copy of the gene for it to be expressed. Heterozygous is where we have big A, little a, for example, so we have 2 different alleles. And then genotype is all of the DNA in an organism that then obviously codes for what that organism is like. So let's start with dominant. We'll work down the statements until we find the one. So the collection of alleles that determines characteristics, no. The alleles that are both the same for one characteristic, no. You only need one copy of this allele to have the characteristic, yes. Heterozygous, the alleles are different for one characteristic, that's good, we've got 2 different alleles, so it's that one. Genotype, the characteristics that an organism has, no. That would be phenotype. So it's been put in there as a bit of a red herring. Your genotype is the collection of alleles that determines your characteristics. Yes, that is true. So just be careful that you know the difference between genotype and phenotype, because that's what they try to do there. Make sure you just eliminate statements that don't make sense. It's not going to be exactly the same as the definition you've learned. Like I said, it will just be what the closest is, and using that process of elimination of what's correct and what's wrong in order to help you find the answer. Part B. In humans, hair colour is controlled by a gene. This gene can either be capital B, which is dominant, or little b, which is recessive. A combination of capital B will give dark coloured hair, brown or black. A combination of little b will give light coloured hair, blonde or red. What colour hair will a combination of capital B, little b give? Okay, so this is just, do we understand? If someone is heterozygous, so they have a capital letter and a little letter, or a dominant allele and a recessive allele, what will the outcome be? What characteristic will they have? Well, they will have the dominant characteristic, because you only need one copy of that allele in order to have that characteristic. So they will have brown or black hair. Orla has red hair. She is currently expecting her first child. Her partner Nadim has brown hair and is known to have the combination of alleles capital B, little b. So he is heterozygous. Use a Punnett square to show the potential genotypes of Orla and Nadim's children. So if Orla has red hair, she must be 2 little b's. And we can already put in Nadim's because it says he is capital B, little b. So we're going to put in the 2 little b's, and then we're going to fill in the Punnett square, which is lovely and nice and easy once you've got the gametes in the right place. we have a capital B and a little b, two little b's, and then exactly the same again on the second row. So this gives us our genotype potentials for our offspring. What's the probability that Orla will give birth to a child with red hair? So now we can use this Punnett square to say which of the offspring of the genotypes are going to produce red hair. So that'll be the two little b's. And there's 2 of those out of the total of 4, which is the probability is then 50% or a half, however you want to write it. Decimal or a percentage or a fraction is fine, as long as it's 50% or a half. If Orla gives birth to a girl, what sex chromosomes will she have? Okay, so this is to do with whether you have XX chromosomes or XY chromosomes. Typically, females have XX chromosomes and males have XY chromosomes, and that's the kind of fact that you were taught as part of the course that you need to know here to answer this question. So if she says that she's having a girl, then this girl should have XX chromosomes. So here's the mark scheme for question 7. We've got dominant, heterozygous and genotype definitions, so you get literally 1 mark for each of those being correct. Dark brown, brown or black, you can have any of those options. Punnett square axes, so getting the little b and the big B, little b right in the top of the Punnett square gets you 1 mark, and then you get 1 mark for having those correct sort of offspring genotypes inside the 4 boxes. You will get 2 marks for all of those being correct. If you got 50% of those incorrect, you can still get 1 mark. Then you need an answer of 50%. As we said, you could say 0.5, you could say a half, or you could say 2/4. If you didn't simplify the fraction, still gets you the mark because it is correct. And then XX for the female sex chromosomes. Okay, on to our final question. The diagram below shows the cross-section of a kidney. Okay, so that cross-section just means you cut them in half to look inside. So we've got a diagram here. I would say, like with all diagrams, if you can, try and label up what you can before you read the questions, because it'll probably help you. So outermost layer is the cortex, layer in the middle is the medulla, and then the very innermost layer is the renal pelvis. So I know all of those. D is the ureter, because that's clearly going down to the bladder. So it's taking urine down there, so that's where urine's going to go. And then the other 2, the renal artery, the renal vein, obviously my blood vessels. So our first question says, which letter represents the medulla? Well, helpfully, we've already labelled that for ourselves, so it must be B. What is the function of the part labelled D? Again, we've already labelled it. It's going to transport the urine to the bladder. It's just, the function, remember, is always just asking what does that part do? So I need to add the word transport here because it transports the urine to the bladder. Part C, describe the method for testing a urine sample for glucose. Okay, so we should have talked about sort of this practical at some point, either in food tests and as part of food tests and testing for glucose, or as part of this topic and talking about urine. So it's basically asking us to describe the method, so practical steps about how you test for glucose. So hopefully we know that you should add the Benedict's reagent, because that is the one that tests for sugars, and make sure you put a capital B because it is named after a person. What do you have to do with it method-wise? Then you have to heat it up. So I would talk about heating it, probably in a water bath, or boiling it in a water bath to get it to work. And then what are we looking for? We're looking for a red-orange precipitate, or the colour change to red-orange for there to be glucose present. So add Benedict's reagent and heat in a water bath to 80 degrees. Urine tests from 3 different patients, so that means obviously they've got numbers rather than just a colour. The normal levels of glucose in the urine is 0 to 0.8 millimoles per litre, and the normal levels of albumin in the urine is 0 to 3 milligrams per millimole. We really need to focus just on the numbers here, don't necessarily worry about those concentrations for now in terms of the units. Albumin is a protein, so we do need to know that, and we should hopefully have recognised that. So we've got a table where we've got 3 patients and we've been told what the normal levels are. So I would go through the table quickly and let's see what we're looking at. Are we looking at normal levels? Are we looking at normal levels of all for all the patients for glucose and albumin, or are there some differences? I'm going to expect some differences because I'm going to expect that they want us to talk about it. So glucose concentration patient 1 is normal, it's less than 0.8. Normal for patient 2, it's less than 0.8. It is above for patient 3, so it's 0.96. Albumin for patient 1 is normal. Albumin for patient 2 is very much not normal. It has very high. And then albumin for patient 3 is normal. So now we've kind of flagged that. We've got that in the back of our minds ready to answer these questions. First part says, what condition is it likely that patient 3 is suffering from? So patient 3 has higher than normal glucose, but normal albumin. So if they've got high glucose in their urine, then that's likely that they will have diabetes. That is a symptom of diabetes. Part 2 says, explain what the results in the table for patient 2 indicate about their kidney function. So patient 2 has normal glucose, but it has higher than normal protein. So we need to explain why high protein, or what high protein could tell us about the function of their kidneys. So how can we link the kidneys and their function to high protein in the blood? So normally proteins in the blood will be quite low, and we know that we've got the levels there because proteins are too big to fit through the filtration system in the kidney. They stay behind because they're too big to pass through. Only small molecules pass through and then end up in the urine. So if there's lots of protein there, it means that that filter has been damaged somehow because it's not stopping those large molecules from breaking through. So that's what we're going to say. We're going to say what the result for the table for patient 2 is, because it doesn't say that in the question. So they have a high protein content in their urine, or high albumin content in their urine, and so it suggests the large proteins that normally too big to pass through have passed through the filter, so it suggests that there's damage to the filter part of the kidney, so there's kidney damage or damage to the kidneys. So last question then on the paper is part E. If a patient has kidney disease, they can be placed on a dialysis machine. However, they may also go on the waiting list for a new donor kidney to undergo a transplant. Discuss the advantages and disadvantages of each type of treatment. Okay, so it's quite a long answer question here. We'll always have at least 1 6 marker on the paper. They always want us to do one long answer. Here's quite nice because they've said that they want advantages and disadvantages of kidney dialysis versus a kidney transplant. So always do some little notes before you start writing this out, because obviously there is this quality of communication mark here. So you are going to need to write it out sort of well and make it logical and make it make sense. And we'll talk about how we do that in a minute. But for first, I would just make sure that you can get at least 1 pro and at least 1 con for both transplants and dialysis as a minimum. Otherwise, we're not going to be able to be hitting those top levels of marks. So the pros, the positives, or the advantages of a transplant is once you've got one, you don't need to be on dialysis anymore. Your kidneys will work. It will also extend your life expectancy because having a functioning kidney is obviously much better than having dialysis, so you will hopefully live a bit longer. The disadvantages of a transplant then is that there can be a really long wait for a donor. You are obviously going to have to undergo quite serious surgery with anaesthetic, and there's a risk there, risk of infection and risk of death from the surgery. Once you have had the transplant, you will be on immunosuppressant drugs for the rest of your life, which can be a bit of a burden or can impact your life because you are then more prone to getting various infections because your immune system has been weakened by these drugs. The benefits of dialysis, it's going to keep you alive or keep the patient alive whilst they're waiting for a donor, which is obviously very important. You're not having any major surgery, so there's much less risk of infection and death. You're still having like a port put in terms of sort of where they can remove the blood and stuff like that. So there is still a risk of infection, but it's much lower, and there's no risk of death from anaesthetic or anything like that. The cons are it can only be a short-term solution. Ultimately, even if you have kidney dialysis, you shouldn't be on it for a certain length of time. After that, your body will still start to fail, so you will eventually still need a transplant. Whilst you're on it, you will have to have some kind of controlled diet, which obviously can affect your life. And if you're going and sitting in the hospital for hours on end, a couple of times a week to have your dialysis, that's a really long period of time where you have to sit in a hospital and you're kind of trapped there. It's going to affect things like your job, your work, and your life with your family and things like that. So it's kind of a sacrifice you have to make to kind of time out of your week where you have to sit still in one place and you're not necessarily, that's going to impact how you live. Okay, so the way I've written this is I've written this as a coherent argument. It's kind of, we're not doing like a paragraph on this and then a paragraph on disadvantages and a paragraph on advantages. I've tried to link them together, which is how we get to that kind of high level marks in these answers. So As I go through, I'll try and sort of make sure you can see where I've put the advantages and the disadvantages. So let's do pros in green and cons in pink. So everywhere where there's a pro, I will underline it or highlight it so we can see the mixture as I go through. So I've been quite clear at the start. The advantages of dialysis are that it will keep a patient alive during the long wait for a donor kidney to be found. It also does not require major surgery, unlike a transplant, so there is less risk of infection or death. So in that statement, I've been able to say that these are the pros of dialysis and compare them to the cons of the transplant, because I've mentioned the long wait and I've mentioned the fact that you have to have major surgery and that there's risk of infection. So I've mentioned both as I was writing. However, dialysis is not a long-term solution, unlike a transplant, which would extend the life expectancy of the patient. A controlled diet and lots of time spent in the hospital for dialysis sessions can impact the life of the patient significantly. However, there's also a lifelong impact with transplant surgery. Although no special diet is needed, they will be on immune suppressant drugs for the rest of their life. Oh, sorry, that is a con. That should be pink. So no special diet is needed. That's a pro, but they will be on immune suppressant drugs for the rest of their life. And then I've added why that's important and that's an impact. It's because it could make them more vulnerable to infections. So you can see here how I've blended my arguments. You don't have to do that. If you wrote this out as bullet points very similar to what we've written on this side, that is fine. We're just not comparing them or discussing them together. You're talking them about as if they were separate things. And you're not maybe making some of those logical links that are going to get us into that top bracket of marks. So for the mark scheme for our last question then, we had B, which was obviously the labelling of the medulla. Transfer or transport urine to the bladder is what the ureter does. You've got your steps for doing a Benedict's test. Main thing here is that you really need to make it clear that you're talking about Benedict's. Do not get this confused with PhET reagent, so the spelling cannot be ambiguous. You really need to have made sure you said it properly. You have to say boil or specify a temperature that is above 75 degrees. Try not just to say heat. It's not really enough. If you warm it up a little bit at a low temperature, that's not going to have the same effect as actually getting it up to a high temperature. And then just be clear about the orange or the brick red. Diabetes is the disease, and the reason is because we've got protein in the urine being higher than normal. Proteins would not normally pass into the nephrons because they would be too big, so it suggests kidney damage. Then we've got our long answer question for 8E. So it's got all of the content that we sort of mentioned and some other things as well on the left-hand side. We didn't really talk about rejection. We talked about the medicine about that, but we didn't talk about it. We didn't talk that the dialysis is obviously costly to the NHS. The fact that you've got all of these on this side, but the main thing is that just writing these isn't necessarily going to get you into that 5-6 mark bracket. It wants you to show sequential reasoning, which is what is the statements that are in bold. So like we said, can't just say, oh, it's major surgery. You have to say, so therefore it risks infection. Like we said, the impact on their life and work about sitting and having dialysis for long periods of time. The fact that you can leave patients vulnerable to communicable diseases if you have those immune suppressants so that they could get more sick with infections. Those are kind of the statements that are not just facts but kind of give reasons that explain why it's a pro or a con, and that is what gets you into that high level mark bracket. Okay, so that's the end of our paper. I hope you found that useful and you've managed to spot some maybe content areas that you might be weak on that you can now go away and do some revision on. Definitely get some more past paper practice in around those topics as well once you've reviewed the content so you can have another go, and make sure you target that revision now to those areas that you found hard in this paper, whether that was content or whether that was the types and styles of exam questions like this long answer question. If you found that hard, then go and find some more long answer questions to practice.
Ouch! This is why in some videos I explain scratches.